Hi everyone. I'm going to go over the colony morphology lab and the pigments of bacteria labs. So these labs are two different labs, but they're very similar. So I'm going to describe them in one video. In this first portion of the lab, you're going to try to determine which bacteria are what specific species by looking at specific morphology or shapes that bacteria have on agar plates. So here in this image, we see bacterial agar plates, and we can see that there's some distinguishing features. So for example, this agar plate has some brown colonies on it. On these agar plates, the colonies are a bit fuzzy. So we're going to talk about that. And then the other portion of the lab is pigments of bacteria lab. So some bacteria produce different specific colors or pigments that distinguish them from other bacteria. And that's what we're going to talk about. I'm going to start off with the colony morphology lab. Colonies are bacteria seen on agar plates. So here's an agar plate. All these little circles are colonies. And what a colony specifically is it's millions of species of bacteria that originated from just one bacteria. So one bacterium was there on that agar plate and it did binary fission millions of times so that we're able to, with our eye, see that circle, which is what we call a colony. So colony is a pure culture of bacteria. It's one bacterium that divided millions of times, so that colony is millions of bacterial species. It's not one. If it was one, we wouldn't be able to see it because remember, bacteria are microscopic. So that little colony circle is millions of bacterial species. And colony morphology is basically what do bacteria look like on an agar plate. So their morphology, their shape on agar plates. It's important to know colony morphology because different bacteria look differently on agar plates, and this way we are able to distinguish them. So as microbiologists, we have knowledge of what different bacteria look like on agar plates, and some bacteria have very key features that make them stand out on an agar plate. So what you're going to do, if you are actually in lab, you're going to look at colony morphologies of 4 different bacterial species and write what is different about them. So how can we distinguish the 4 different bacteria from each other? The bacterial cultures you are going to look at were Bacillus glaubgii, Bacillus licheniformis, Bacillus megaterium, and Bacillus mycoides. And all these bacteria look very different on agar plates. So we're going to describe what they look like. And for each bacteria, we're going to look at that bacteria grown at 22 degrees Celsius, which is room temperature, or 37 degrees Celsius, which is body temperature. So here's Bacillus glaubgii. Bacillus glaubgii, here is bacteria growing at 22 degrees Celsius. Here's the bacteria growing at 37 degrees Celsius. I noticed that the bacteria grows at both temperatures, and at both temperatures, it's an orange brownish color, but at 37 degrees Celsius, at the higher temperature, the bacteria likes its temperature more, so we see more growth at that temperature, and it's also more brown. So this is Bacillus glaubgii. It has very distinctive color and look at both temperatures. Here's Bacillus licheniformis. We don't see any growth at 22 degrees Celsius, but the bacteria does grow at 37 degrees Celsius, and it forms these very big colonies. Here's Bacillus megaterium. It forms these nice round colonies, and it grows at both 22 degrees Celsius and 37 degrees Celsius, and I would just say they're white colonies. Bacillus mycoides, you can tell it looks very different than all the other bacteria. For me, I would describe it as like a fuzzy looking bacteria, and it does grow both at 22 degrees Celsius and 37 degrees Celsius. We see more growth at 22 degrees Celsius of Bacillus mycoides. So if I had all the bacteria given to me and I saw plates that looked very fuzzy, I would automatically know that they're Bacillus mycoides. So that was the morphology lab. Now we're going to talk about pigments of bacteria. So some bacteria produce pigments, which are just in essence colors that they produce. And these pigments can have very different functions. They can protect bacteria. They can be to do photosynthesis for those bacteria that are photosynthetic, such as cyanobacteria. They can be antioxidants for the bacteria. So there's so many different functions that the pigments do for the bacteria, and just know that they're byproducts of their metabolism. So you guys learned about metabolism. Sometimes they'll produce a pigment just as a side product of their metabolism, and that pigment can go to protect them. So pigments can also be intracellular or extracellular. Intracellular means that the bacteria produce it and it stays inside. Extracellular means that they release the pigment to their environment. The pigment and the intensity of the pigment can change based on different environmental conditions. So maybe at one temperature, we see one color, and at another temperature, we see another color. Or maybe at one temperature, the pigment is more intense, like it's more red or more purple. And as well, it can also change based on the media or what we grow the bacteria in. The pigment and the intensity of the pigment can change if we have light or no light. It can change if you have oxygen or no oxygen, so in aerobic versus anaerobic environments. So keep that in mind. So we're going to go through a lot of bacteria, and I want everyone to memorize what the bacteria look like at both temperatures and what pigments are produced. So let's go through. Okay, so here's Bacillus globgi. Both of these are pictures of the bacterium Bacillus globgi. Here it is grown at 22 degrees Celsius and 37. This is the same bacteria, but these ones these tubes were incubated longer, so we got better colors. So if we look at this bacteria, it grows at both temperatures. At 22 degrees Celsius, it's orange. At 37, it's a much more intense pigment, and it's a little bit like dark orange brown. So that's what Bacillus globgi looks like. Here's Chromobacterium violacum at 22 degrees and 37 degrees. At both temperatures, this bacteria produces a violet or purple pigment. Here is Micrococcus luteus at 22 degrees and 37 degrees. It grows at both temperatures and it looks yellow. The pigment that it produces is a yellowish color at both temperatures. Here's Micrococcus roseus, and this bacterium is a rosy pink color at both temperatures. We see a stronger, more intense color at 37 degrees Celsius. So at the higher temperature, the bacteria produces a more intense rosy pigment. Here's Pseudomonas aeruginosa, which is very common in different infections such as lung infections in hospitals. This bacteria, it has a yellowish growth at 22 degrees Celsius, and then at 37 degrees Celsius, so at body temperature, we see this nice strong green color. This is important because it helps us distinguish Pseudomonas from other bacteria. So if a patient has a potential Pseudomonas infection and you take a sample and you send it to lab and the lab tech streaks it and they see this bright color at 37 degrees Celsius, they right away know that it's a Pseudomonas aeruginosa infection. Here's Serratia bacteria. Serratia bacteria at 22 degrees Celsius, so at the lower temperature, is a red color, and then at 37 degrees Celsius is just this clear white color. Here is Xanthobacterium lividum, and this is a violet color at 22 degrees Celsius, and it does not grow at 37 degrees Celsius. Unlike Chromobacterium violacum, which grew at both 22 and 37 degrees Celsius and is purple at both temperatures. Here's Sarracenia. This bacterium grows at both 22 and 37 degrees Celsius, and it's an orange color. And then finally, here's Rhodospirillum rubrum, and we grew this bacteria in the presence of light and no light, as well as aerobically and anaerobically, so with oxygen and with no oxygen. And we noticed that with the bacteria that's grown with light, they form a pigment, which makes sense because when you have light, bacteria would create a pigment as a form of protection. And then when there's no light, the bacteria are colorless. And this light bacteria were grown anaerobically, no oxygen, and no light bacteria were grown aerobically. So from both of these, the main thing I want everyone to get is I want everyone to know what are the distinguishing features of all the bacteria we talked about, like the colony morphology of Bacillus megaterium, Bacillus mycoides, all the bacteria we talked about, the pigments of Serratia marcescens, Xanthobacteria lividum, all of them. Know what they look like. If I give you a picture of anything, know exactly what bacteria it is based on the pictures. Know that colony morphology and pigments of bacteria are important because this is how we distinguish specific bacterial cultures. Know that some bacteria grow differently at different temperatures. We see different colony morphologies, different colors. And finally, I want everyone to know why bacteria would form pigments. So we talked about it. Know that it can be a form of protection. It can be for photosynthesis. It can be for antioxidants. And they could protect bacteria against solar radiation. So just like our skin forms melanin, so it forms the pigment melanin when we're out in the sun to protect us against skin cancer. Bacteria also produce pigments to protect them, so it's a form of protection. So go through all of these and hopefully all of it makes sense. Know what the distinguishing features of all the bacteria we talked about.